Hello everyone, today we're going to figure out which RNA viruses are the most cursed, and we'll do it in the form of a tier list, because who doesn't like tier lists? Tier lists are fun. Alright, first of all, let's make a little bit of space here. So first things first, to rate their cursedness, we'll mostly consider things that are like related to their life cycle, like how they work, any kinds of properties that you would generally consider unusual in virology. All of this is, of course, perfectly subjective. If you disagree with any of the ratings, feel free to comment down below. Also, these are in no particular order, just to go through them really quick. So we have enterovirus, we have rabies virus, we have influenza, we have measles, we have corona, HDV, that is hepatitis D virus, dengue, HIV, and noro. And we'll try to stay roughly within like a minute per virus, so this doesn't exceed 10 minutes by too much. Alright, so let's jump right into it. So to set the stage a little bit, it might make sense to start with something that is not very cursed at all, to give you sort of a baseline. So why don't we just start with the enterovirus over here? So these are pretty much as standard as you can get for like a plus-strand RNA virus. I mean, the genome is basically mRNA, makes a big polyprotein as you would expect, which is then cleaved by protease into all the individual proteins that it needs. One of them, of course, being like an RDRP, like an RNA-dependent RNA polymerase, which will then make more of it and will reassemble into new virions, which are, of course, not enveloped. So yeah, is there anything unusual about it? I mean, it has this interesting thing where it doesn't really disassemble the whole capsid. It basically threads the genome into the cytoplasm from the endosome, so that's pretty cool. It is one of those viruses that use a VPG protein as a 5' cap, which is not very stealthy. So basically, instead of a real 5' cap, it will have like a little protein that it attaches, and that can also initiate translation. But yeah, the cells will not appreciate that. It's like a very non-stealthy way of doing things, which is fine. This thing just wants to come in, replicate really quickly, and then basically blows the cell up to get out. Yeah, so is there anything else about this one? I mean, the taxonomy is pretty messy. It has like a whole bunch of different potential manifestations that it can take. I mean, polio is an enterovirus. If you want to read something a little bit unsettling, you have to look up, I think it's called encephalitis lethargica. It was like a weird outbreak that happened in shortly after World War I, I think. Also called the sleeping sickness, and I think people nowadays assume that it was pretty likely an enterovirus that caused it. So yeah, if something like that happened today, certainly would be a little bit scary. But yeah, I mean, all things considered, this thing is pretty much as standard as it can get, right? So I certainly wouldn't consider it cursed in any way, so we'll just put it down here in the F tier. Okay, which one shall we take next? Let's do corona. Yeah, so I think everyone had a little bit of exposure to this recently. But is there anything notable about coronaviruses on the biology side? I mean, for one thing, they are like really big. They're like among, or probably the biggest RNA viruses that we know of. I mean, they're a plus-strand RNA virus. There is this proofreading function that they have that makes it possible for them to actually be that large. Like copying RNA is usually a pretty sloppy process. The nidovirales are basically the only ones who actually have a proofreading function for their RNA polymerase, which is a pretty unique feature. Okay, anything else? I mean, so they do this thing with the polyprotein, but they also make subgenomic RNA to be able to have a little bit more control over what is made in what quantities. I mean, that's like one of the bigger disadvantages if you go for a big polyprotein. For every translation run, you literally just get one of each. and you might want a lot more of certain things than of others, right? So the subgenomic RNA method makes a lot of sense. Okay, anything else? There is certainly a point to be made about this whole idea of like host switching. This is not very common among viruses in general. So coronaviruses, and I mean, we've seen this with SARS-CoV-2 a lot. SARS-CoV-2 is known to infect a whole bunch of different animal species. I mean, the mink and the white-tailed deer come to mind, but I think it can also infect cats. The ability to jump into different species gives you the option to do like mutations that would not be possible in your other host species and evolve this way. Also, you can potentially do recombinations. I mean, the whole reason SARS-CoV-2 like came into existence was probably like a recombination event at the end of the day. So that's pretty unusual. Okay, anything else? I mean, it is pretty strong in the conspiracy theory department. I mean, where are we gonna rate this? I mean, it would be cool to put it into C for Corona, but I think we'll just settle for D tier. I mean, it's better than it's definitely more cursed than an enterovirus, but it really doesn't hold a candle to some of the other ones we have here. So I think D tier is fair. Okay, next up, let's go further down the respiratory route with influenza. So this is the first negative strand that we have, and it's also like, I think, the only segmented that we have in the list this time. Influenza is notable for like a couple of different, not unique properties, but unusual and things that like undoubtedly make it very effective at what it does. So I mean, let's look at the obvious thing first. I mean, this whole idea of being segmented, with influenza having 8 different segments, kind of allows you to just swap individual parts of the genome around. That gives you the ability to like evolve parts of it in a different context and then maybe use it again in the original context or in a completely different context. Influenza actually does replicate in the nucleus for an RNA virus. That is very unusual, but it also gives them access to like this cap snatching mechanism. So we basically have all the different cap functions within our first 3 picks. Enterovirus with the VPG approach, Corona with the we synthesize our own caps, and then we have influenza, which basically takes a normal mRNA, cuts off the cap, and then uses it as a primer. So it like its RNAs, its mRNAs literally look like host mRNAs because they are derived from host mRNAs. So yeah, what else? I mean, influenza is one of the top candidates for like really severe outbreak scenarios. The sheer diversity and the ability to recombine, in quotation marks, with different strains. So this whole antigen drift, antigen shift mechanism has a lot of potential. The question is, would we call this an A-tier cursed virus? Look, it literally replicates in the nucleus as an RNA virus. I think it should be placed at A. Like, this thing is pretty weird. Okay, what next? Oh, let's do measles. Okay, so first up, like from the replication strategy perspective, this thing follows a very standard mononegavirales strategy. If we look it up on Viral Zone really quick, it's pretty much exactly what you would expect from like a paramyxo. It's a negative sense genome. And of course, for that to work, you also have to include a polymerase in the virion. The polymerase also handles the capping and everything. And I mean, if we take a look at the replication cycle over here, it's a very short list of items that happen. It's a very simple strategy that you can see in a similar way, for example, in RSV and in many other mononegavirales type viruses. Okay, so why is measles special? So measles has a couple of really bizarre properties. It basically starts the infection in the upper respiratory tract, then causes a systemic infection to then come back into the lung. So this is one of the few airborne types of viruses. I think the R0 number is something between 15 and 18. So each infected individual on average would infect 15 to 18 others. It's one of them, like it is among the most contagious viruses that we know. Also has some drawbacks, of course. I mean, RSV, you can have that pretty much every year. With measles, it's like a one and done, right? Once you have antibodies against it, the systemic infection part will not work anymore. So it's pretty much a one time only for the most part. Okay, yeah, but this is pretty unusual. Is there anything else? I mean, it has this whole thing with the truncated M gene. So what it can do is transition into like a form that doesn't even produce virions anymore, and it usually does that in the CNS, and it can then lie dormant for years before basically reactivating and causing like a rapid encephalitis that is then also usually lethal. This is what's known as the SSPE, subacute sclerosing panencephalitis. Pretty rare occurrence, but very spectacular when it does happen. But does it warrant an A tier? It feels like an A tier virus to me, I have to admit. Maybe we have to bump it down a little bit for being so boring in the replication department. Also it has like, it is very strong in the conspiracy theories. It's basically like the OG conspiracy theory. Maybe that warrants putting it into A tier. Yeah, let's put it into A tier. If we are counting conspiracy theories, maybe we should also bump up the coronas a little bit. Okay, what else? Yeah, let's do HDV. Okay, hepatitis delta. What is so special about hepatitis delta? Some of you might have heard that it can only exist as a co-infection with hepatitis B. But yeah, this goes a little bit deeper. So if we take a look at what this thing actually is, like it is a single-stranded circular RNA at the size of 1.7 kilobases, which is basically nothing. I mean, we've talked about the other RNA viruses. With Entro, that would be around like 8,000 or 9,000. The Paramuxos are, I think, around 15,000 if I remember correctly. This is only a tenth, like you will hardly get more than like a single gene on this. If we take a look at the one protein that it actually encodes, you will see that it is some form of nucleocapsid in a way, or nucleoprotein. It doesn't have a polymerase. So how would it replicate if it doesn't have a polymerase as an RNA virus? Well, it actually does it by using the host RNA transcription machinery. Specifically, the RNA pol2 does it, which is extremely bizarre. This is something that simply is never done, right? There are things called viroids in plants, which are actually apparently not completely uncommon there. These look fairly similar in the way like they are set up. They're also circular single-stranded RNAs with very complex secondary structures, just like this one. But the viroids generally do not encode for proteins. This thing does encode for a protein, which makes it even weirder. And it is reliant on the HBV surface proteins to turn it into infectious particles. It's one of the weirdest things I've ever come across. So this one definitely has to go into S tier. It's like, this is basically the most cursed thing in virology that you will ever see. Okay, dengue. Don't even quite remember why I included this one here. I mean, it's a flavivirus. Has like it has a very standard replication strategy. Again, I mean, it's a plus strand genome. It makes a polyprotein and cleaves it basically. Just here, it's a little bit more complicated. It does have like a capping function contrary to the enteroviruses. But otherwise, I think it's the only vector-borne virus in here if you don't count rabies. But yeah, I mean, other than that, like, is there anything else about it that I'm missing right now? Well, we'll go right into F tier then, I guess. Okay, moving on. Rabies. This is a cheerful one. So this is again one of those that follow a pretty standard mononegavirales strategy. It has this very interesting bullet-shaped virion. But other than that, like, it's a lot of things that we already saw in the measles, right? But what makes this thing special is really its propagation method, if you want. So what will basically happen is the virus is in the saliva, and it will then do a local infection where you got bitten. It will start infecting axons of neurons that are innervating this area on the motoric side. So it always goes retrograde, right? It just follows the axons in a retrograde direction, which will, from the motor neurons, lead you eventually into the brain. From there, it will then go further retrograde into like the sensory part, which will get you into the salivatory glands, among other things. And it will then also cause a non-inflammatory encephalitis, which is always lethal. And it does cause this famous phenomenon of hydrophobia. And if you take like a look at a rabid dog, like the increased aggression, like the foam on the mouth, like the virus is in the foam basically, which is saliva. It cannot swallow the saliva because the throat muscles are paralyzed, so it will drip out. And at the same time, it will be afraid of water because if it tries to drink water, it will know that it cannot swallow it. And yeah, it's probably one of the inspirations for all the zombie virus movies. So yeah, I mean, from like the replication side, it is very mundane. But yeah, I mean, I've made presentations about this and literally had nightmares afterwards. So I'll put this at least at B tier. Okay, Noro. Why did I put this on here again? I mean, it is a Kaliki virus, of which there are a couple. It's very similar, or like it has a lot of similarities with the enteroviruses. It also uses this VPG strategy for capping. It's one of these diarrhea viruses, and it's really good at that. No, I mean, this one is pretty standard. Like, it has a lot of parallels to the enterovirus. It does make some sort of subgenomic RNA, apparently, with VP1, which would be the capsid. Otherwise, it makes a polyprotein. Yeah, I mean, I don't know. It's not very spectacular on this area. I can remember having it once. Basically couldn't get out of the toilet for around 6 hours. One thing I do remember is that for some reason I couldn't drink coffee afterwards for multiple weeks. Like the smell alone was unbearable. And I have no idea how I survived that, because no coffee is usually lethal. Okay, but where do we put it? I think it's just F tier, right? I mean, it's not super interesting, or at least it is not cursed in any way that I can see right now. Which brings us to HIV. Okay, this one you probably all know. The good old human immunodeficiency virus, the agent of AIDS. It is an RNA virus, but it has a very deviant replication strategy. It's actually like, it's kind of a little bit in a class of its own. It's not really an RNA virus like the other ones here are. So what it does is it basically goes through a DNA intermediate. It can reverse transcribe its own RNA to then integrate it into the chromosomal DNA, making it a provirus. And the provirus is actually where you then transcribe the actual genes from. Also has a pretty interesting capsid just on the side here. Yeah, but how cursed is that actually on the cursedness scale? Retroviruses are kind of rare as in human disease, at least nowadays, but we know that there have been like ancient retroviruses in the germline. I mean, we can literally find the genes that are in the human genome right now that are part of defunct retrovirus genomes. You also find it in pigs and a whole bunch of other mammals. So retroviruses really aren't all that uncommon. But still, I mean, the strategy is pretty wild, like, have to admit. It's like a little bit convoluted. I mean, if you look at if you compare like all the steps that you need to make or to replicate HIV compared to the Mononega viralis, which had like 5. So this is like a needlessly convoluted way of doing things. Does it warrant an S tier rating though? I mean, it can't be lower than A. I think it's fair to give it an S tier rating. Like, yes, I think that's reasonable. So we have our 2 S tier viruses, HDV and HIV. A tier with influenza and measles, B with rabies, C with corona, and then a whole bunch in F tier, which are usually just the run-of-the-mill plus-strand RNA viruses. But yes, there you have it. Yeah, so I hope you enjoyed this little video. Apologies for the crude presentation, and totally didn't take enough time to go into any of this in any detail, but yeah, if you're interested in any of them, just let me know in the comments, and if there are any other tier list topics that you would also like to see, yeah, just put that down below as well. So thank you very much for watching, and see you next time.